It's an honor to introduce our next speaker, Rohin Mhatre. You may know or may not know that graduated with PhD here, Barnett Institute. Ira, are you somewhere in there under Ira's? There we are. So we're so proud to have one of our own, been a good friend to Northeastern in the interim. Started 20 years at Biogen, 20 years at Biogen, somewhere in the middle, already very influential, but by the end, pretty amazing, senior vice president with a long title that meant was in charge of just about everything having to do with large molecules, proteins, development, process, analytical. I mean, it's really amazing. And now Rohin is the executive vice president, excuse me, and chief technical operations officer at Fog Pharma. It's my pleasure. Thanks.
Good afternoon. So nice to see you folks, all. Pick up? Okay. That better? Okay, good afternoon. What I say, not as nice to see you. No, I said so nice to see you all. But, it's particularly, I sort of think back of the days of the Institute. I still remember the courses I took with you guys, with Barry, you, Paul, you, Ira, And I was in Ira's lab. I, graduated after some interesting work, a little bit of therapy, and, it all worked out okay at the end, Yeah. But
it's group therapy.
Yeah, it's group therapy. So, I'm excited to be back, and to see so many of my colleagues here that were at the Institute, and some of them that are. It really was a very interesting time, the time that I joined. Biotech was, up and coming. And at the same time, there were some really incredible things happening at the Institute, which were very impactful to sort of the growth of this industry. So before I start, I do want to congratulate everyone, the faculty, staff, all the students that are here, Barry, you for the early days of getting this institute going, and the Barnett family. It was truly a very unique setup, and it continues to be so. Very few places where there was such a focus on bioanalysis. And also, while that was happening, what helped us all as students was this whole collaboration across the industry. We were collaborating with all the leading pharma and biotech companies at that time. And that was really something, once we sort of graduated, it really hit us because it gave you an exposure as a student as to what reality was, And that really was a very big thing. There's a number of projects that came out of it, publications that came out of it, but it sort of prepared you in a very different way than, some of my other peers that had joined the industry. It was a realization of, what applied research meant and, what was the industry looking at, and it allowed us to do a lot of interesting things as we sort of stepped in. So I was fortunate to join the biotech industry. I started my career with Applied Perceptive Biosystems at that time, and then joined Biogen. And it was a unique time for this industry. I'll sort of start with, where biotech was in the 90s. Clearly there were a number of established, by relatively speaking, companies that had launched some products, but none of them had quite the portfolios of what they have today. I remember when I joined Biogen, we had one product that was launched, and that was it. It was Avonex, and was nowhere close to it. The industry was finding its footing. There was a reasonable amount of funding and so on. But at the same time, the modality was primarily peptides and proteins. It was mainly proteins. And for many of us that sort of joined, the whole technology was not quite what it is, particularly the ways of making these molecules, the way of analyzing them was nowhere close to what it is now, and it continues to progress. Like biomanufacturing was a very inefficient process then. It was very, very inefficient. The cost of manufacturing was extremely high. There was limited understanding in terms of what all this meant, what what actually impacted the product. There was all this discussion about, cell culture as a black box and so on. There wasn't as much understanding as to what really impacted the productivity, what impacted the quality of these products. And interestingly enough, two of the techniques which were so influential in this field, CE, capillary electrophoresis, and mass spectrometry, thanks to many of you folks that are here, were sort of considered exotic, for the lack of a better term. And there are a number of folks from the Institute, I see Wasim and Stacy, who helped sort of the whole area of CE flourish, brought that to the industry, brought that to regulators and so on. And likewise, the same thing with mass spectrometry. So a lot of good work that came from this institute was quite instrumental in sort of moving some of these boundaries as far as getting a better understanding of product quality assessments. So I think we all need to feel good about that, of some of the impacts that we've been able to make in the industry. And at the same time, where the analytical became so important was this whole idea, everyone was talking about well-characterized biopharmaceutical. I mean, it was still relative in terms of what well-characterized meant then, 20, 25 years ago, to what it does now. So that's really where it was. And fortunately, this industry has progressed, and quite in very significant ways. There have been dozens and dozens of very effective therapies that have been licensed, blockbuster drugs, the Enbrels and the Humira, Avastins, and so on. And it continues to be so. But what's interesting is, a biotech company as we knew it 25 years ago is very different than what it is now, where almost all the players, when you look at sort of what the traditional biotechs were, a large amount of their portfolio is small molecules. And many of the traditional pharmas that had happened, they sort of went into proteins, and you have many of the large pharmas in gene and cell therapy. So it's all sort of a diversified portfolio, but interestingly, through all of that has been a lot of joint learning. There have been things that we did with proteins. We are trying to implement that for both gene and cell therapy. It's kind of in terms of how we're producing it. It's getting to more protein-like production systems. There's need for increasing the productivity just in terms of the indications that we're looking at. And also what's really important, and something I want to touch upon today, is that has piqued my interest for several years, is the opportunity for end-to-end sort of a digital transformation of this business. And there are many sort of experts here today. I saw some of the panels, so you probably know more of this than I ever will, but I speak of it from a real interest in and some of the work that I've seen already coming out of it. I think it's sort of transformational. Sort of one has to look at sort of what's the next thing coming in, and it's really the end-to-end possibilities of a digital transformation. So somebody had sort of shared this slide with me. I bring this up because the reason why this industry has always evolved, because despite all the success, there are a number of sort of factors that influence its growth or sort of prompt it to innovate constantly. There are always, despite the amount of revenues folks have been able to generate over the years and so on, there's always a fine balance. It is getting more and more expensive to bring drugs to the market, to launch them. The expectations have increased quite significantly. Manufacturing costs are high. What we're producing, some of the indications that we're looking at, needs a large investment. And to a large extent, it's investment at risk. So it's a changing landscape. The whole regulatory landscape has changed. How do we bring these products to the market? How do we supply them? The supply chains have become far more complex. And COVID, unfortunately, was a true test of that, where there were significant impacts. But just the diversity of products that are coming out, the way we're launching it, going for global launches, it's a very complicated sort of system. And all these things coming together, the whole digital tools become extremely important in all of this. And a number of companies have been fairly successful in getting that together. This just gives you a picture when I talked about the end-to-end. When you're looking at digital tools, one of the areas that I see, and that's very relevant, I bring this up to a number of students that may be looking into this. I think there's an opportunity from the small molecule industry where they started doing a lot of in silico type of work, and something I'm very keen on. I started some of this at Biogen, but even as I'm transitioned to Fog, this is something that's been a big commitment, is really starting to look at how do you bring vast amounts of data that's available today, pulling that together to be able to do a lot of predictive type of work, moving away from wet labs. This is not to say to shut them down or anything like that, but trying to kind of get into a state whereby you can leverage all this data, all this historical knowledge in this industry to be able to start looking at, whether that be a synthetic pathway, that has been happening in the pharma industry for a while, but likewise performing a lot of in silico work to be able to develop conditions and processes and so on. And this goes all the way. It's sort of looking at how do you control processes, how do you look at product disposition, product supply, and even basic things. To give you an example, I was on the production floor not too long ago, and one of the things, a simple thing as a pump that's been used in for pumping, fluids here through the process. What something like a digital tool can do, where we were looking at simple vibrations of a pump, and based on those vibrations, being able to predict what the life cycle would be. When would you have valves on a pump go away? And these are impactful things. This may sound sort of a very minor thing, but are very impactful because it's looking at sort of, issues on the production floor and so on. It's just an example of sort of how you can bring in predictive tools. And I'm urging a number of companies that I've talked to and academic institutions, particularly here, to sort of continue this emphasis on the digital evolution as much as you can. My interest in the industry, whether the days of Biogen and what I'm doing now, I think overall, to do all these things, we have to sort of figure out a way to build a culture of innovation. And for the industry, it's particularly important with the kind of disease states and all that we're looking into. And one of the things that we've been trying to do, both in my previous company and now, is getting better at predicting what the business needs are. When I was sort of working earlier in gene and cell therapy, I sort of was indicating the patient populations are growing up, the larger and larger indications that we're getting into. So how do you get into a state where you can start predicting outcomes? And this is something that has been of interest to me. And also starting to think about overall, as an innovation culture, that innovation is not really all about technology and fancy tools. And for this industry to progress, and for a number of upcoming fields right now, is really trying to envision possibilities. And I think that's really is a good way to think about it. And we've been trying to do that. I'll go over that in a minute. Some of the goals that we set have to be ambitious, because when you start looking at it, to move away from this, low-hanging fruit type of an approach. So the process that we've been trying to establish for a while, once you sort of have that basic culture of innovation, but it's to set really ambitious goals. I'm in the business right now where we produce peptides. It's primarily for oncology indications. And as some of you may know, this is a very expensive production process. It's highly inefficient, and when you look at cost of production, it can cost up to about $4,000 a gram to make some of these therapeutic peptides. These are all made solid phase. So when you look at something like that, we've been sort of looking at the innovation journey, is trying to set more ambitious targets. To a large part, we want to decrease the cost of goods at least 20-fold to make this a viable sort of setup. And there are a number of tools available to do that. Obviously including a lot of analytical technology that would be necessary. So I'm just sort of giving you a particular example of that. And what we have been able to do for a while, a number of folks, Elaine, I don't know, you remember Biogen days, and even now, is to sort of set up tech investment programs, which is how we started collaborating with the Institute. A number of collaborations, Barry, if you remember over the years, it really came from this, where there was seed money to start looking at all the possibilities. These were blue sky ideas, and you just have to go at it from a point of not sort of looking at sort of safe destinations. So that's sort of where things landed up. And the opportunities for efficiency right now as an industry is a number of these platforms are evolving. I mean, I look at new modalities. I mean, look at oligonucleotides, peptides, gene and cell therapy, and so on. And there is an opportunity for a lot of reliance, as I said, on these digital tools. An area that has been of particular interest is the real utilization of cloud labs. Because if you're truly looking at broadening your base in terms of how you can get the work done and sort of leveraging infrastructure, the cloud lab has become a very important thing. And I'll touch upon some of that in a minute. So this is just to give you a sense, from in the digital age, you can start all the way from the beginning, envisioning what you're trying to treat. But another important indication and another important application, rather, of this is the patient. And the digital tools that are coming out these days in terms of adherence, monitoring, and so on, disease progression, helping better adherence to the drugs is just remarkable. And it's several separate talks on a topic like that. But this is what I'm hinting at, where we were able to get to in my previous job and where we're heading to even my current operation, is really to look at what is a high-performance operation. And to a large part, there has to be a significant level of automation built into that. And what that would mean is basic operations that are so standardized that you could actually run these things. I mean, to give you an example, and folks, mass spectrometrists here would appreciate that. When I started my career, as I said, it was a highly sort of, you need very, very skilled individuals to kind of run these instruments and so on. And not to say that you don't need those folks anymore, but operating the most sophisticated instruments at this stage, Amar, you would remember that. We had sort of had recipe operations for this, where you could have somebody running the most sophisticated techniques. Clearly, they may not have been able to interpret a lot of this data, but just in terms of basic execution, it was a matter of a few days in which you could get the most sophisticated instruments run across the board. It's just simple examples like that. When we're looking at manufacturing operations, a lot of decisions are being made on the floor thanks to all kinds of analytical instruments. And I kind of think this is important because much of that effort is going on here as well. And then I'll talk a little bit in terms of what that would mean in terms of product disposition. So this is really what processes look like, what we are aiming towards also at my present company. And that is get to a state where, with all the analytical tools and technology that you have, you start with even basic characterization of raw materials. Get to a state where you fully understand what these inputs are. And then an important element, all of this on the manufacturing floor, is predictive control. Being able to read very quickly what's happening in reactors, what's happening in reaction vessels, and being able to influence the outcome of your process. So you have consistent input going in, you have consistent control. So the idea then would be whatever comes out, your product in this case, is as consistent in quality as one would imagine. And that's this whole idea of predictive control and advanced manufacturing sort of aspects. So the general approach here is we've built very clear connections in terms of all the process parameters that affect product quality. Predictive models that then control the quality, and then process signatures. I mean, simple things like a peak shape or a growth curve of a cell can help you make a lot of predictions in terms of what a manufacturing process is going to be. So all these tools essentially are what's giving us an idea of product quality and consistency. And as I had said, modeling becomes a very critical tool here, right from the get-go, and modeling on all kinds of different things, looking at facility modeling, looking at process modeling, looking at mixing, heat transfers, all those things. I mean, a simple thing like a facility modeling can have such a significant impact on the cost of your production, because interestingly, in manufacturing, one of the things is not how much you make, is how much you can move through your facility. And so when we use terms like volumetric productivity and so on, I think that's what it is. And a lot of these models help you figure out what is the best flow of all your or your processes and all your product through your facility. So it's a very different world than when I started. You know, moved away from all paper-based systems. It's gone all electronic, whether that be for manufacturing batch records, looking at quality systems, and so on. It's a different and an exciting world. And when we look at modeling, you know, as I said, a number of you may be far more familiar with this than I am, but we've looked at it from very basic things. You know, I mean, when you start looking at modeling what would be a good raw material, you know, complex raw materials, you know, building learning sets, you know, incorporating some of the machine learning through that, then gradually including PAT sensors, trying to look at sort of predictions, and then eventually looking at what an entire predictive model would be. So this is a good amount of work. You know, when I talk about modeling, it's not somebody going in there, obviously, as you know. But many of these things that we have been able to put into our facilities, it's a few years of work, and not all of this is guaranteed to work. So, but when it does, I mean, the impact is quite significant. You know. So this is, I just want to give you a sense of what this would mean. I'll just pull out a couple of things here. So when I talk about some of these models, this is a classic example, you know, in a bioreactor setup. You're using very basic tools, Raman spectroscopy, which has been around for decades at this point. So these are a number of probes that are in bioreactors, and you're constantly reading, whether that be glucose, lactate, dissolved oxygen, you know, all kinds of components in your feeds and so on. It depends on how far you want to go. And this is just giving you an example of sort of how this works. So what you see on that side, on the extreme right here, is monitoring glucose, you know, where you're looking at just complex spectra. This is not sort of looking at what is a spectra for glucose, but you're looking for changes in spectra as you're generating training sets. This is when you are increasing glucose levels. And what that helps you do is then predict online, you know, through a trace or basic trace from a Raman spectra, what would be the level of glucose or any other component. And then through a feedback control, which I'll sort of show you in a minute, this is what it would look like in a manufacturing. This is being implemented for some time now in my previous company. So this was a very basic thing where there are constant reads of what's going on real time in your process. You know, so if you read a glucose level that's exceeding or is decreased by a certain amount, you would have automated loops that are feeding all this information. You know? And it's moved away from things like a lot of bolus feeds that were done. So what this has enabled us to do is really get to very consistent cell growth and obviously product quality. So if you take this is an entire integrated operation which is based in Switzerland. This was through Biogen. And what that has allowed you to do is through the entire process looking at right from raw materials all the way to the final bottling of a product, a number of tools that actually help you constantly monitor quality. So every unit operation of your production cycle is where you can read with basic instruments. You know, some cases they're sophisticated instruments, but in a lot of cases they're basic instruments which help you read what the product quality is and being able to make decisions. You know? So it's a constant automated feedback loop. You know? This is just sort of showing you how glucose is monitored. I won't spend much time on this, but the impact of this is then on glycosylation of your molecule. But this is telling you, these lines here, the blue and the black lines show you how a predictive model versus an offline measurement, and the correlations are extremely good, you know, as you start sort of, as the model matures. To take this a step further, even in purification, you know, one of the big bottlenecks you have is aggregation. So how do you constantly assure low levels of aggregates? And it's a very simple thing where you're reading through this, through your purification, you know, what aggregation looks like, and then you're making decisions on the floor, how much to load the next column so that you get consistent aggregate profiles. And, you know, in some cases we've gone rather sophisticated where you're reading out of a bioreactor using, you know, sampling loops, 2D systems doing LCMS, doing a lot of detailed product characterization through bioreactors, throughout the bioreactor cycle, and so on. So it's a number of folks here that have been quite instrumental in this, Lee and others that have been, in this and have generated some very interesting data over the years. This is just, again, another trace out of a manufacturing flow. This is looking at a certain component. And constantly, the red lines sort of show you what the standard deviations are. The green line is the average, and the blue lines is what you can do in terms of control. So it's a constant control of all these components. So you're never at a stage where you have variability any more than what you need. And this is what eventually it could look like, where this has been put in practice to a large part. You're moving away from testing everything on the floor, in a lab versus moving things on the floor, predicting what product could look like, and just going in a very different frame of where this industry has been. So, anyways, I hope this kind of gives you a little bit of a perspective of where things are going and what is the future. I mean, I wish I would be that good in predicting, but nonetheless, I think sort of it is going to be a very different sort of state for the industry, the folks coming in, the students that you can train. I hope sort of it eventually gets to a state where the different skills that come into the industry than what have been, and I think it's much needed. So thank you for your attention, and again, thanks for the invitation.
So when you think at a university now, somebody who wants to get a PhD in analytical chemistry, who is either going to go into an academic position or industry, what are a few of the courses or areas that would be new or from when you were there or when I started?
Oh, good point.
So that might be an interesting one.
So
for the
it
people.
would have to be more of I would say, a mixed curriculum,
That doesn't say anything.
No, exactly, it doesn't. But similar to things, better understanding of aspects of things like, machine learning and other elements of AI. More savvy in terms of some of these modeling type of experiments that I showed you. Some of this changed because we started recruiting folks that came out with this background. And even in my job right now, I have people working on process predictions that have no idea about chemistry. So it's, obviously it would be much helpful, but many of the skills are not with the chemists that are coming out these days. So those are types of skills that that hybrid would be an ideal approach, because that's not there right now.
Of the problems you described, which one or something else is the worst?
I don't know if it's the worst, but there are several areas where we don't quite have processes to make the products that are going to be required, the scale at which is going to be required, and that the cost of production is too high. So which can have a significant impact in terms of supporting these indications. I don't know if that answers your question, Roger. But no, don't give him the microphone.
He was one of my PhD students.
Oh, geez.
Yeah.
He was probably the best PhD student I ever had. Don't let him fool you.
Okay.
He's extremely clever.
One question only.
Question. There's a world of difference, as you know, between small molecule chemistry and big molecule chemistry. Ira closer.
Never had a problem with that.
And what you've described seems to me to be easily applicable to small molecule pharmaceuticals, but it may be much less applicable to biopharmaceuticals, which, as you know, are much more complex.
Yeah.
How do you propose to go from one to the other?
The two separate tools, right? For the pharmaceutical industry, when you're looking at reaction routes and so on, there are a number of sort of tools available that have matured over the years, right? But the same thing in the biotech. I mean, there are production platforms. So you have to be able to leverage that. So some of the in silico work that we were doing, we're looking at production platforms work that we had done. I mean, at this point, when you look at, many of these companies, you've developed hundreds of products, and the platforms have matured to a point where you don't need to go into doing all the lab work. I mean, basic purification. There's so much known in terms of the impact, PI of a protein, aspects of solubility, aspects of binding, and so on. So, I mean, we haven't leveraged that. And I think the one of the issues also is data accessibility. So while I'm sort of saying this, the other big investment that's happening in companies across, I mean, there's not just biotech, is really all the sort of the data integration that needs to come in, which is a very expensive proposition.
Just thinking about analytics in the bioreactor, you mentioned Raman and glucose monitor temperature. Like, what else would you measure to make the predictions better? Like, what is the analytics that we don't do now and would make it easier?
So in some
Do you have mass
cases,
spec in the bioreactor or what?
yeah. So it doesn't all have to be in the bioreactor. I mean, some of these probes are relatively straightforward, right? But a lot of it can be sampling. Done sort of sampling, many of these proteins, you can clean them up, protein A, I mean, they're rapid cleanup processes. And then you can go into more sophisticated analysis too, looking at product quality. I mean, things like glycosylation. There's no way to change that glyco profile once it's formed in the reactor. None of the purification is going to impact it in any significant manner. So it's really getting a better sense of what you're generating. Now, I'm not saying that these are all mandatory, because even as these models have been developed, as the processes mature, what has happened is we would then reduce the level of monitoring that's needed, because you have better sense, and there are indirect measures of figuring out what the product quality would be. All right, thank you.